Ever wondered how basketball players grow so tall? Or how that cut on your hand just healed? The answer? Mitosis. So before we get started, there's a few things that we need to know already. First of all, what is DNA? Second of all, what are chromosomes? And third of all, how is DNA replicated? If you're not sure about any of this stuff, check out the links below in the description for videos I've made on all three of these questions. So let's start with a definition for mitosis. Mitosis is a part of the cell cycle where one parent cell divides into two daughter cells, each with the same number and type of chromosomes as the parent cell had. Or put more simply, a type of cell division that creates identical copies of cells. Thanks, Toto. Okay, so based on the definition we've been given, we know we're going to start off with one parent cell, and we're going to end up with two daughter cells. Now this process can be split up into 1, 2, 3, 4, 5 steps. And we're now going to go through on a pretty high level how we go from one parent cell to two identical daughter cells. The first stage is called prophase. When we look inside the cell, we can see two chromosomes. There's a blue chromosome on the right and a red chromosome on the left. From my other videos, you'll know that this is a homologous pair. In this case, let's say the blue chromosome is from the mother and the red one is from the father. We also know from the fact that the chromosomes have this X shape that the DNA has been duplicated. Again, see the links below for other videos that help explain this. At this stage, we can start to see the outside of the nucleus is starting to break down. The next stage is metaphase, and you'll notice that the chromosomes have moved so that they're now lining up one on top of each other along what we call the equator, which is the middle of the cell. This is an important step for the next phase, anaphase. In anaphase, you'll see that we've now pulled these chromosomes apart. From my previous videos, you'll know that each side of the chromosome is called a chromatid. So here, we've literally pulled the chromosome apart and we've taken the chromatids to opposite sides of the cell. In telophase, we take this one step further. We continue to move the sister chromatids to opposite sides of the cell. And now you can see that black dotted line, which is the same as the one we had before. We're starting to form a new nucleus around each of these separated chromatids. Finally, in cytokinesis, we're going to split this large cell in half. We'll end up with two new cells, and notice the nucleus is fully formed around each of these new chromosomes. Now there is one added detail that we need to cover. You'll see I've now added some purple lines, and these are called microtubules. These are special proteins that help line up the chromosomes along the equator of the cell during metaphase. In anaphase, they also help pull these chromosomes apart. And they do that by searching for the centromere in the middle of the chromosome and then pulling on that. That's what helps drag each of the sister chromatids to the opposite poles. We call each of these the poles, and they're on either side of the cell. The other thing you may have noticed is that the daughter cells at the end of mitosis aren't exactly like the parent cell at the beginning of prophase. And we said in our definition that mitosis creates two identical daughter cells from the parent cell. So we're missing some information here. Now, in order to understand that, we're going to have a quick look at the cell cycle. So here we have it, the cell cycle. We can see it's a cycle because it cycles around. If we start over here at mitosis, this diagram and these diagrams should look familiar as the first stage prophase of mitosis and the final stage cytokinesis that's even labelled here where we have two cells. Now the first stage of the cell cycle after cytokinesis will just follow one cell. This cell will go off and have its own cell cycle off somewhere else. So if we follow this cell, we can see that the first stage of the cell cycle is G1, which is cell growth. And during this phase, the cell grows and everything excluding chromosomes will be duplicated. You'll notice that the cell at this stage looks exactly like it did earlier because we're focusing here on the chromosomes. The next stage is called S, which is synthesis. During the synthesis stage, you'll notice that the diagram goes from having one red line and one blue line to essentially two red lines and two blue lines. And the reason that's happened is because DNA has been replicated during the S phase. After DNA replication has occurred and we go from having a single chromosome to an X-shaped chromosome that's got two sister chromatids, we need to go through the G2 phase which checks the duplication. Make sure that the DNA replication that we did before is actually correct. So during G2, all the way up until mitosis, we're just making sure all of that's been done properly. Once we're happy, we can take this cell, go through mitosis, and you end up with 2 new cells. Something important to note here is that in a normal cell, we have 46 chromosomes. Now, when you look at these diagrams, you can't see 46 chromosomes. You can only see one. One's come from the mother and one's come from the father. The reason we do that is because if I had to draw 46 of these different lines, the diagram would become very confusing. So we only look at one homologous pair. Now remember, the homologous pair, one's come from the mother and one's come from the father. So in fact, there would be 23 of the red chromosomes and 23 of the blue ones. 23 plus 23 equals 46, which gets us to the 46 we want. So as I say, in every normal body cell, we also call those somatic cells, there are 46 chromosomes. And what we really mean by that are, there are 23 pairs. 23 chromosomes have come from the mother and 23 have come from the father. Now ultimately, we want to take that one cell that's got 46 chromosomes and make exact copies of it. We want to create 2 daughter cells. And if we look all the way around the cell cycle, you can see that we actually end up with 2 cells that look exactly like this. And so these must have 46 chromosomes in each of them. So we've successfully done mitosis. However, when you look at this cell, you can see that something else has happened. And as we know, in the S phase, DNA replication has taken place. What this actually means is that if we look at this cell, we don't have 46 chromosomes. We've duplicated them. So in fact, for a period of time, this cell has 92 chromosomes. We go all the way around, after we've checked the duplication, we still have 92 chromosomes. But once we've split that 92 in half, if you divide 92 by 2, you end up with 46 again. Well done, nearly there. Last thing to note is just the purpose of mitosis. So as we talked about right at the beginning, we can use mitosis to make identical daughter cells for growth, for repair. Also, if you're looking at this from the point of view of a single-celled organism, by going through mitosis, you can go from one single-celled organism into 2. And in that way, you can go through asexual reproduction. If you want more information on asexual reproduction, check the links in the description below for a link to another video all about asexual reproduction. Finally, mitosis is also important for genetic stability because, as we know, it creates identical daughter cells. They have the same number of chromosomes as the parent cell, and so they have some role in genetic stability. So there we have it. I hope you enjoyed the video. Please like and subscribe if you want to see more. See you next time.